The overall goal of this procedure is to isolate single cells for microarray-based transcriptome analysis. This is accomplished by first dissecting the retina from the surrounding tissue and then dissociating it into single cells. The second step is to harvest single cells and deposit them into PCR tubes containing lysis buffer. Then the cells are lysed and the cDNA is amplified using reverse transcription followed by polymerase chain reaction. Ultimately, hybridization of the single-cell cDNA on an Affymatrix microarray is used to reveal the mRNAs expressed in a single cell.
The main advantage of profiling the gene expression in single cells versus using whole tissue approaches is that single-cell isolation allows you to gain access to very small populations of cells that would comprise the entire tissue. These cells could be anything from different types of neurons to vanishingly small populations of stem cells. What the single-cell technique allows us to do for these cells is to identify both cell-type specific markers for these small populations of cells and to identify gene networks that are expressed in these extremely rare subsets of cells. Additionally, the single-cell profiling technique allows us to identify the gene networks that are transiently expressed during very dynamic developmental transitions in very small subsets of cells.
This method can help to answer key questions in the field of developmental genetics, such as when progenitor cells begin to express gene networks associated with differentiation, and how those progenitor cells differ from one another. The implications of this technique extend toward cellular therapies for neurodegenerative diseases such as glaucoma, because by elucidating the key networks in the development of individual cells, we're one step closer to being able to generate those cells for use in cellular